Alright, this is your video on the PCR lab. This is the food safety lab. So what we have is an E. coli outbreak that is on the space colonization program, and we have one of two choices for the contaminant food. We have an interhemorrhagic E. coli that is either found in the space burgers or they're found in the space sushi. So what we're going to do is we're going to run a PCR and run a gel to find out which food source has the pathogenic bacteria. We're going to amplify a region of DNA that the pathogenic E. coli actually has a restriction site for XMN1. And therefore, when we run the digestion and then run the gel should have two bands for a pathogenic species or pathogenic sample, and then only one band for the non-pathogenic sample. So, how this works, this is the timetable for the PCR lab. The reagents and equipment will be at your stations ready to go. You're going to set up the PCR, which is mainly going to be filling the tubes. Now, PCR tubes are a little smaller than the microtubes you're used to. We've been working with 1.5 milliliter or maybe even 2 milliliter tubes, but also we've been using a lot of 500 microliter tubes or half mil tubes. But you're actually going to use 200 microliter tubes or tubes that are a fifth of a mil for max capacity. So the PCR tubes are much smaller. So the programming will take about 5 minutes, and honestly I'll try to expedite that process by having it already set up, and all you'll have to do is load the tubes and then run the protocol, which will take about 40 minutes, 30 to 40 minutes. Now, after you have loaded the tubes and have run the PCR, we're going to run a digest on the amplified regions. So after all of the PCR has been concluded, right, and the protocol is finished, you can either digest that day, which, like I said, you'll have the enzyme ready to go. You add it in there, put it at 37 degrees and in the water bath, and let it go for 15 minutes. Or you can leave that for next time and then just be ready to start with your digestion at the beginning of the period in the next lab. Okay, and then of course the last bit of the lab is to run the electrophoresis. Now, if we're stopping after the digest, we're going to heat inactivate at 65 degrees. That will deactivate XMN1, and then we'll store it in the freezer. If we don't, if we do the stopping point before the restriction digest, then we'll just put straight in the freezer. But I'll have both the 37 degree water bath for the actual digest, and I'll also have a 65 degree water bath for the heat inactivation of the restriction enzyme. Like I said, then we'll run the gel and then determine the size of the bands and interpret which food source is the pathogenic food source, or contains the pathogenic organism. So here's your setup. You're going to have 4 yellow PCR tubes, or they could be white or blue or green depending on which ones I've dished out. You're going to have them labeled A, B, P, and NP, so you'll need to label those. And in A is where you're going to put your space sushi DNA. In tube B is where you're going to put your space burger DNA. In tube P is where you're going to put the pathogenic E. coli sample, so that's your control. And then in the NP tube, you're going to have the non-pathogenic control. So you'll know what a pathogenic species looks like, or a pathogenic sample, and a non-pathogenic sample looks like, and we'll do a comparison. You should also have your tubes labeled with initials or group numbers on the side so we can identify who's who, because in each PCR thermocycler, we're going to have 2 groups in the thermocycler at a time. So your PCR reagents, what you're going to be adding to each PCR tube is template DNA. So 5 microliters of your tube A, or sorry, of sample A will go into tube A, and then 5 microliters of sample B will go into tube B. 5 microliters of the control P will go into tube P, and then 5 microliters of control NP, which will go into tube NP. But you'll have primer mix that contains all of the primers for attachment, and then you'll have PCR master mix, which contains ions, and it contains Taq polymerase, as well as the deoxyribonucleotides used to make the double-stranded DNA. So your end volume here is going to be 30 microliters. Now, I would suggest that you go in a different order. Not go 5, 10, and 15. I would probably pipette the larger volume first, and then do your smaller volumes as you go. So it'd be my recommendation to go 1, 2, 3 in that order. Okay? And also, be sure to change the tips after each step. You're going to gently mix the reagents by pipetting them up and down. Do this 3 or 4 times, and then you're going to cap the tubes. Make sure all the liquid is at the bottom of the tube. We can spin them down in a microfuge to get all the solution down there, but you can also gently tap them on the table, and that will get everything back down there as well. You're going to place the tubes inside the thermocycler, and then I'll show you in class how to close the box and secure it so the lid won't come off and it will work properly. So your PCR programming, you're going to have a Chromebook or a laptop or even your phone. You can actually run right off your phone with the proper adapter, which is kind of cool because it makes this process very mobile. But you're going to run a predetermined protocol, which will be preset. It'll already be up. I'm going to make it easy on you guys. So what you're going to have is you're going to have your 5 steps. The first step is the initial denaturation, where the DNA is going to open up, and then is going to, so here's your initial denaturation right here. That's really horrible. Let's go ahead and try a different color. Let's go blue. There's your natural denaturation, which is kind of hard to see in this, but it's 94 degrees at for 30 seconds. Okay, so that 30 seconds is going to open up the DNA at a high temperature, and then we start this cycle right here of heating up, cooling down, and then extending our DNA primers once they're being made through the process of PCR. So our initial denaturation, and then we enter up another denaturation state, which is at the same temperature, 94 degrees for 5 seconds. And then we have annealing, which is the attachment of the primers, and then that's going to be at 57 degrees for 5 seconds, and then the temperature goes back up to 72 degrees for 5 seconds to cause the extension of the process. So the actual DNA replication is occurring at that point from the primer to the end of the sequence. And then after 30 cycles of the red box that I just drew right there, of 94, 57, 72, 94, 57, 72, we'll do that 30 times. And every single time that we open up the, denature the DNA, we're going to get the possibility for orders of magnitude more copies of DNA, specifically the fragment that we want. And then when it's all said and done, it's going to have a final extension, which will be another 72 degrees for 30 seconds. Okay, but that's the whole PCR process. And like I said, this whole thing should take about 30 to 40 minutes. Okay, moving on. Once the PCR is completed, make sure you let it cool because it is warm to the touch, and actually be a little light on the thermocycler saying that the lid is hot, so you're going to want to wait until that, or not on the thermocycler, but on the computer screen to say that it's warm, you may want to wait until the lid hot indicator turns off. And then once it does, you can remove your tubes. But in the meantime, while you're waiting for those tubes to cool, or even while the PCR is running, you need to label 4 other tubes, which need to be labeled AX, BX, PX, and NPX. And what this is trying to show is this is going to put the same substance that's in your PCR tubes. You're actually going to split that into 2 aliquots. One's going to be a digest, the other one is not going to be digested. So, like it says, because you have right now 30 microliters in your PCR tubes, you're going to take half of that, so 15 microliters, and you're going to put 15 of A in the AX tube, 15 of B in the BX tube, 15 of P in the PX tube, and 15 of NP in the NPX tube. So as of right now, you're going to have 8 tubes, right? Each one will have 15 microliters. Now, at this very moment, they are going to be identical. But this very next step is we're going to add a restriction enzyme to all the X tubes. So AX, BX, PX, and NPX. Now, one little change we need to make right here is because you guys are using P20s, your P20 micropipette, you're not going to be able to accurately pull out 1 microliter. So I've diluted it, so you are going to be adding, right, 2 microliters of the restriction enzyme MXN1 or XMN1 into each of your 4 tubes. Again, using a different tip every time. So you're going to pipette the restriction enzyme directly into the PCR product. That is the AX, BX, PX, and NPX. Pipette up and down to mix it well, and then cap the tubes to ensure that the liquid is going to be at the bottom. Okay, if we got to spin it down, then we'll spin it down. But understand that there is a chance that your PCR tubes might be in a strip of 4. And a strip of 4 PCR tubes is not going to be able to be run through the centrifuge. So you may want to just be careful and make sure you're transferring everything into the bottoms of the tubes. So the digest runs for 15 minutes in a water bath. There is a protocol to have a heat block protocol for your PCR tubes, which you are welcome to do. But like I said, I'll have the water bath going either way. And then you're going to run a gel. I will have a 2% gel already made with the appropriate number of wells, and these are the lanes that each group is going to run. So there's going to be 8 groups. We're going to have 4 gels, and then it's going to be 2 combs per gel. Okay, but lane 1 through 9 are going to be the same for each group. So in lane 1, you're going to have 100 microliter of a 100 base pair ladder. Lane 2 is going to be 12 microliters of A. 3 is going to be B. 4, P, 5, NP. Okay, if you notice, they're all 12 microliters. And then in 6 through 9, you're going to have the digest of A, B, P, and NP, respectively. So there is no need to, like it says right here, to add loading dye, because everything, the PCR master mix that you added to the PCR tubes and the ladder are already load ready, so you can just load them straight in there and watch the gel. The gel itself should take about 15 to 20 minutes to run. Okay, we might stretch it as fast or as, go as long as 20-25 at a lower voltage, but we're going to have it set for 100 volts, and we're going to shoot for about 20 minutes. Okay, if you guys have any questions, then either leave a comment or ask in class.